This is my personal theory about how to get to immortality, how it'll work, or perhaps what we can actually spend research efforts into and get there. So some of it is going to be fairly sort of research focused, but I've tried to make it as practical as I can. So there's this. So in day one, we talked about the theory behind what this longevity stuff is, and then also why we die, why aging happens in the first place, and I talked about the small molecule approach to fixing things, which is find some pill or chemical or whatever that helps. And then the second thing is Aubrey de Grey's SENS approach, this idea that a bunch of different types of junk builds up, and then if we just go in and fix all this junk, then we can basically live forever, because that's the sum total of what goes wrong. And then the third thing that I talked about in the first day was the idea of injecting order into the body. So I begin with some practical advice. Frankly, all this is pretty obvious. That we all sort of know healthy living when we see it. Important to have a good doctor. If you have health issues, deal with them. There are various interventions like stem cell therapy, FMT, that are kind of coming online. And this is all stuff that we talked about before. So there's this phrase, you are the average of the 5 people that you spend the most time with. And I think there's a lot of truth to that. And so if you want to make longevity a serious goal, then you may want to focus on surrounding yourself with people with a lot of knowledge and with a lot of practical skill and application. I think that there's going to be a lot of sort of secret knowledge floating around in the future about, for example, FMT, stem cell therapy, and various sort of odd therapies. And this knowledge is not going to necessarily be evenly distributed. And by surrounding yourself with people who really keep up to date on this stuff, you can learn about what works, what's important, before it gets big. So I would say, other than air filters, this is one of the best pieces of advice. But the core focus of this talk is going to be about far future, or not necessarily far future, but technology that doesn't just slow down aging, it doesn't just extend your life, but could actually stop aging, could actually literally reverse aging. I mean, it's the big prize in longevity. And as I was arguing the first day, 1/3 of a sort of a utopia. Super longevity, super happiness, super intelligence. So, how do you do it? First of all, I just want to point out that our capacity to edit our genomes is increasing fairly rapidly. That there's this technology called CRISPR that so the backstory here is that scientists were looking at this bacteria in yogurt, and it had a very strange genetic sequence, and a lot of repeating things and some old virus code, and they were trying to figure out what it did. And it turned out that it's this crazy system for precision cutting of DNA. And it's basically this bacteria's immune system that it can cut up viruses, and with this CRISPR system, just slice them in two. So the scientists so another group of scientists figured out that, oh wow, you could actually build a gene editing system with this, because you can sort of target which sequences to cut, and then you can cut them in a fairly precise way, and then have your own chunk of DNA in to sort of integrate in to where you cut, and then it ties it back together, and then you have a new sequence. So amazing use of nature's kind of innate technology. So that's it's pretty new, and people, I mean, it is the hot thing in biology. Everyone's working on it. So it's rapidly improving. There's also this idea of whole chromosome synthesis that we're getting better and better at printing longer and longer strands of DNA. George Church's H.G. Wright, I think, is probably the best example of this. Basically, if we can print off a strand of DNA as long as a human chromosome, then we can just make new human DNA. And we don't have to edit it. So these I think are the most exciting two threads in gene editing. There's also viral vectors, which are kind of older technologies, and iterated embryo selection, which is sort of a way to choose genes for your children. Anyway, the bottom line here is that we'll soon be able to create cells with basically any DNA we want. And it may not be today, it may not be tomorrow, but that future is definitely coming. But the problem is we're sort of building all these powerful things, but we don't know what to do with them. We have these amazing scientific tools in our toolbox now, and they're getting better and better and better, but we still don't understand the genome well enough to muck with it at a very deep level. There's this, people talk about genetic engineering, but that is not the era that we're in. We're in the era of genetic tinkering. We're pre-paradigmatic here. Engineering implies a precision of effect that we simply don't have. However, so there are sort of four ways to overcome this problem. And small text here, don't worry about it. But basically, 4 ways to find good genes to edit. And the first is you just take statistics and say, okay, this gene is pretty much correlated with being tall, being healthy, being smart, whatever. Maybe it's a good gene. This is, again, statistics, the worst form of argument. But there are other approaches, like let's take a gene from jellyfish and put it in this apple, maybe something cool will happen. And this is done. It's
smart apple.
Yes. And the third is to sort of try to piece together building blocks of biology artificially. And this is sort of developing, but all these I don't think are very much ready for prime time. I think by far the coolest idea here is fixing genetic typos. So And this is what it means in humans. That everyone has a lot of broken genes. On average, we all have about 100 broken genes out of 20,000 or so. And my 100 will be different than your 100, will be different than yours, and yours, and yours. On average, we get about 50 of these broken genes from our mom, 50 from our dad. We cleanse about 2 based on low fertility differential reproduction stuff. And then we pick up about 2 based on just the copying errors of making a new person. So it stays at about 100. And there's also, so that's one way to look at things. These broken genes just aren't making any proteins. So the purpose of genes is to make things. These genes aren't making anything. So they're just broken code just hanging out. And yes. There's also and so this way takes a look at completely broken genes, but there's also genes that are associated with poor health. So using IQ as a proxy. And we have about 1,000 of those, that 1,000 sort of minor dings in our genome. So basically, we have a lot of stuff wrong with us, is the point. And even healthy people have lots of stuff wrong. And a lot of the things that you might kind of be struggling with might be due to these broken genes. And their effects often get sort of worse and worse over time. So, roughly speaking, we can measure the effects of these genes through measuring their effect on IQ. Not that IQ is this wonderful thing that explains everything, but anything that decreases IQ tends to also decrease health. Anything that decreases health tends to decrease IQ. So it's a tightly coupled proxy. So the basic idea here is, okay, so everyone has a lot of spelling errors in their genome. Maybe let's think about fixing these typos, fixing the spelling errors. This is not something that evolution can do, because evolution is blind. But it's something that if we can actually make precision edits in our genomes, then we can do. And I mean, one sort of heuristic here for typos is, if you have a room of 1,000 people and you're the only one that has a difference in a specific part of your genome, you have a certain version of a gene, and the other 999 people have the other version. Probably the other version is good. The other version is kind of the good version. So that would be a rough heuristic. So there's this question of, yeah, we have a bunch of genetic typos, but it doesn't matter. We seem to be pretty healthy, and how do we know this stuff matters? Steve Hsu from University of Michigan and BGI, Beijing Genomics Institute, has this statistical model of typos where, and this isn't counting this is only counting sort of half these broken genes, but for every 1,000 sort of minor typos that you have, you lose about 1 standard deviation of IQ, 15. And so for every 40 typos you have, and you have about 1,000, so just running the numbers here, if you have about 1,000 of these minor typos, and for every 40 you have, you lose about a standard deviation, 15 points of IQ. Maybe somebody that's really smart only has 900 typos. The point is, what would happen if you fixed all of them? So, just running the numbers here, you would. So, just using IQ is once again a proxy. The smartest person alive has roughly 200 IQ, although this is something that loses validity at the extremes. If you just fixed all your typos, you'd end up with almost 500 IQ. So, first of all, this is a nonsense number in that we don't know what would happen. We don't know for sure this would do that or what that would mean. But what's important about this is it's a sign that maybe there's something interesting here. Maybe there is some low-hanging fruit. And the key thing is we don't have to understand what these typos are doing, how they're bad, in order to fix them. We don't need any more genetic breakthroughs in knowledge, in systems biology or anything like that. If we just have a good heuristic for identifying what is a typo and what is not, and a method for fixing the typos, then we can basically do all this right now. So there's this quote that I really liked about this. What would a spelling checked person, one with no genetic typos, be like? Since no such person has ever existed, we have to speculate. I figure that kind of guy would win the decathlon, steal your shirt and your girl, and you still couldn't help but liking him. So I did a little thinking in terms of using IQ as the benchmark of it's sort of a big target. So half of all our genes are expressed in the brain. So if we have a typo, it probably influences the brain as well. So using, I reduced this super high estimate down considerably to adjust for uncertainty. And then I evaluated all these things for how large of a mutational target each trait is relative to IQ. So I don't know if you can see this, but these are the traits: IQ, slowing aging, beauty, etc. And this is the estimate of how much fixing all typos might improve each trait. So IQ, I estimated, I'm trying to be a little bit more conservative than Steve Shu, so I estimated 5 standard deviations, which would take you from like the average of 100 to an average of 175, which not bad. Slowing aging, I would say, is also about 5 standard deviations of improvement. Beauty, 4 standard deviations. General health, 5. Mental health, 4. Athleticism, 3. Resistance to disease, 2. Happiness set point, 2. Fertility, actually 6. And cancer resistance, 5. And so I would say what this tells us is that broken genes matter for a lot of things, and that if we do have a lot of spelling errors in our genome, it's not just about intelligence, it's not just about longevity, It's about IQ, slowing aging, beauty, general health, mental health, athleticism, resistance to disease, happiness, fertility, cancer resistance. A few things. Basically, literally everything we care about. And there's this idea that, okay, maybe there's this potential intervention, this potential low-hanging fruit that we're finally able to reach out and pick with CRISPR, with precision genetic editing, that we just simply haven't had access to in the past. And so there's this idea that, we talk about CRISPR and, what does it mean? Maybe we could eventually give ourselves new traits or whatever. And I think this sort of frame completely misses the point, completely misses the promise of what could be at stake here, which is enormous benefit on basically every, I put it that every polygenic trait we care about, every trait that depends on a lot of different genes, which is sort of most traits. So there are some research and practical challenges here. First of all, just getting better and better at CRISPR, and people are working on that very, very hard. There's a lot of good people going into that. How do we unify these different conceptions of genetic load of typos? We have the broken loss of function mutations, which have to be sort of analyzed one by one because they're too rare to understand what they do. They're too rare to understand statistically. And then there's the variants that are common enough to understand statistically. We have only a few of these totally broken genes, and many of these sort of minor bad genes. So we need a frame that can unify these two different things. How do we detect these trade-offs? Very important. And how do we sort of make this an easy automated process? Right now, it takes a lot of researcher hours to go through a single genome, but it's just a software problem. So to get to the point, you ask, is this only for babies, for adults? I don't want to say anything about CRISPR babies. That's kind of a hot topic that I wouldn't want to weigh in on at this point. I would just say I hope people understand what is at stake, but not necessarily. I'm not saying that this would be good or bad for society. My focus today is on this, so fixing genetic typos as a longevity strategy. That, okay, we can't go back in time and sort of fix all our typos as an embryo, and frankly, it's much harder to do editing inside the body than it is outside in the lab. So what does this even mean? Can it actually help people, or is this more for can it help people who are alive? And I think the answer is yes, and here's my answer to that. Any cellular therapy, any therapy which involves taking cells out of the body and then putting them back in, and I'm thinking of stem cell therapy specifically here, you can just add a spell check step to all this. So right now with stem cell therapy, we would remove some of your stem cells and then inject them back in where you have an injury. And then the stem cells would move to the site of the injury and help repair things and whatnot. Of course, stem cells degrade over time; they get less effective. So it's questionable how good this would be. But if we took out your stem cells and then spell checked them with CRISPR and then put them back in, you would have the best stem cells that have ever existed in the history of the Earth. And they would probably do a very good job at whatever they're going to do. Likewise, if we're getting better all the time at sort of growing new organs, so culturing a liver or a kidney in the lab, why not make it a spell-checked liver or spell-checked kidney? Or if we want to sort of grow new bone marrow, why not make it spell-checked bone marrow? Basically, instead of getting a transplanted kidney, or even growing a new kidney based on your DNA, that would be about as old as you are, we could basically give you a new kidney or a new liver that is literally the best kidney or liver that has ever existed in the history of the world by far. Mm-hmm. And the question then is, so what? So you have a new liver, so you have a new kidney, so you have new stem cells. Would it matter? And I think the answer to that is strongly yes. That it's not only about your liver, kidney, heart, whatnot. It's about the system as a whole, the overall noise of the system, and this goes up as you age. And this is basically a way to radically improve the signal-to-noise ratio of all your systems. That you have this sort of injecting order into chaos. That if you have a problem, it stresses every other part of your body, and all the rest of your body has to compensate for this. And this, of course, gets worse and worse as you age, and you develop more problems. But if you can go in reverse, if you can remove one stressed part of your body and put in a new version of that is even better than it was when it was brand new, that would remove stress from all your systems of your body. And so, frankly, I think getting a new liver, a spell-checked liver, getting spell-checked stem cells, getting a spell-checked heart, it would allow you to run faster. It would allow you to digest food better. And I would say that I think it will be relevant within our lifetimes. There are these two very powerful forces of development on CRISPR, precision editing, and then development on whole chromosome DNA synthesis, basically just printing off a new genetic code that can be put into cells. And it's unclear sort of which of these will get there first, but they're both improving very rapidly. So this is also dependent on politics and what is seen as acceptable and what is seen as unacceptable. I would say that if you had $1 million to spend on this and good connections, you could maybe make something interesting happen in this space within 3 years. If you had $10 million, maybe 8 to 12 years. Probably timelines will be longer, since most people basically almost almost no one sees this possible future. And frankly, the future doesn't build itself. Somebody actually has to do it. That's it.
Thank you.